So a wrap up, again, as I started in the beginning, in reality, the distance in this multidimensional space that you reduced, so PCA, TSNE, UMAP, ICA, and diffusion maps, will in general reflect these differences in the transcriptional landscape, so the differentiation process, processing the differentiation in the tissue or infiltration in the tissue, and not really the actual time that is spent for that cell. So you really need to have a continuum of cell and really work to have these intermediate cell states and think about this when in the project before doing the sequencing, for example. You make work with a single time point if you have all the differentiation states. So for example, if you take samples from the bone marrow, most of the intermediate cell states are usually there. So even if you just take one sample, you probably will be able to find trajectories, as well as for in some organoids as well, you can also do, or in the epithelium cells, they usually have many intermediate cell state types already in there in the data set. So it is possible to do even with one single sample. But then you also need to know that the intermediate cell states are there. And then you need to identify them first and see, okay, so then my data set is okay to do trajectories. And again, be aware that any data set can be forced into trajectories without any biological meaning. You can, like also mentioned, in a tissue, and you have immune cells. Immune cells do not develop in the tissue, so they are developed in the bone marrow. So any trajectory that you fit with immune cells and together with the tissue, epithelial cells and macrophages and let's say, what do you say, fat cells, it will be completely nonsense. It doesn't mean anything. So you need to take, for example, only epithelial cells and do trajectory on those. And also make sure that the gene sets that you're using have all the heterogeneity for defining these trajectories, and choose an appropriate dimensional interreduction. So don't try to separate your clusters. You can always run dimensional interreduction again specifically for trajectory. You can also run clustering specifically for doing trajectories, because in many cases, for example, oh, I have my progenitor cells and they are separated in the UMAP, so I can identify them. But later for trajectory, maybe you want to fine tune so that all the intermediate states are connected. And as well, you can define into smaller cluster of intermediate cells. So then the trajectory becomes more linear rather than having 3 only clusters. And you'll see that, I think, also in the as well for the exercises. So how do I know which method should I use? So in this paper and in these guidelines for Dyniverse, it is very easy to see. So for example, do I expect multiple disconnected trajectories? So if yes, these multiple disconnected trajectories will be, for example, in a complex tissue where fibroblasts, epithelial cells, and immune cells, they have completely different trajectories. So each one should be fit with a different method. And then there is only one method that you can use. Otherwise, you can just subcluster, for example, epithelial cells, and do the trajectory using these other methods that I'll mention now. So do you expect any particular topology? If yes, then you can try to find, let's say, like linear. If you're modeling cell cycle, then use a cell cycle model. Or if no, then you can go for a different trajectory. If I already know there will be cycles or not, then you can go for trees example or any other graph. And again, I cannot enforce enough. You need to know a lot about biology before fitting curves in here. And in the same paper, they also have measured the overall accuracy, scalability, or usability, if it's user-friendly or not. Some of the graph methods listed here. So for example, PAGA, which is done in Python, and Slingshot, which is done in R, and which trajectories they can fit. So for example, Slingshot will not fit cycle trajectories, but will fit most of the other ones, especially the tree, which is one of the most common found in biology. And PAGA will fit basically everything because it's based on a graph. But in R, race ID, in my opinion, is really hard to use. So as you can see also in here, and Slingshot in their methods or in their comparison is one of the best together with Paga. So, and that's also one of the reasons why we are teaching them here today.